Hello students, welcome back. Today I am Anuradha from Art and Progressive School going to start another chapter, that is chapter number 14. Okay, chapter 14 is respiration in plants. So in the earlier chapter, in chapter 13, we have done about the photosynthesis. Okay, so what was the role of photosynthesis? To synthesize the food, and we have done that there will be two main steps, light reaction as well as dark reaction, right? So in the light reaction, what we have covered, that there will be formation of ADP as well as the NADPH, right? So this ADP and NADPH, if I will just recall it, so this was the thylakoid. So in the thylakoid membrane, what was happening? The light reaction. So in this light reaction, what happened? The ADP and NADPH, it will move towards the stroma of the mesophyll cells. Okay, so here what we are talking about, the stroma of the particular cell organelle, and that cell organelle is chloroplast, and chloroplast is present where? In the mesophyll cells. Okay, so all this ADP and NADPH, when they will move towards the stroma, what happens? It will combine with CO2 which is coming from outside. Okay, so from the atmosphere CO2 will come, and with the help of the CO2 as well as the ADP and NADPH which is coming here, they will form what? They will form the C6H12O6, and this is our main thing which is happening in the photosynthesis. So what is the role of photosynthesis? To make the sugar. Okay, and we know that in plants they are stored in which form? In form of starch. Okay, so with the help of the cycles, that is C3 cycle as well as C4 cycle, we have covered that there will be synthesis of glucose in case of plants due to the photosynthesis process, right? So in this chapter we will discuss that whatever the food it has been prepared by the photosynthesis process, it should be break down, because we know that all the parts of plant, they are not able to synthesize their own food. Okay, only the green parts they are able to do the process of photosynthesis. So the rest of the part, what they are doing, they are utilizing this glucose, from where? From the green part to the non-green part, and those non-green part needs what? Oxidation of this glucose for giving the energy, because most of the activities which has been done in case of plant as well as the human beings, okay, this respiration also takes place in case of human beings also, or in any of the animals. So what is the criteria? Whatever the glucose it has been synthesized by the plant, it should be broken down. When it will get broken down, it will release energy. Okay, so the oxidation of glucose, during the oxidation of glucose, what happens? There will be first thing, it will break down into the smaller unit as well as they are giving the energy. So in this chapter we're going to start about the same thing, that is the respiration in case of plants. It means that how the oxidation of this glucose takes place. Okay, now if I am talking about here the respiration in plant, so we know that in case of plant there will be the cellular respiration. Okay, so what do you understand by the cellular respiration? It means that it takes place inside the cell, and in the common respiration, what was the process? It's just opposite to the photosynthesis. In case of photosynthesis, with the help of what? With the help of carbon dioxide, water, okay, in presence of light and chlorophyll, what? There will be synthesis of C6H12O6, right, as well as the release of water molecule. So here what is the C6H12O6? But this is the product which we are getting from the photosynthesis. If I will reverse this equation, okay, if there will be oxidation, now what will happen? This C6H12O6, it will take the oxygen, okay, it will take oxygen and get break down into the CO2 and water. Okay, so in case of human also, when we are taking oxygen from outside, we are breaking down our food and releasing the CO2, right? So same thing it happens in case of plants also, that the breakdown of this glucose molecule into the CO2 and water. Okay, photosynthesis is the we can say that photosynthesis is an anabolic process. Why? Because there will be a formation of, we can say it's a constructive unit. Why? Because from the simple one it is forming the complex one. But in case of respiration, what happens? It is a catabolic process. Why? Because it's a destructive unit. The glucose molecule will get broken down into the simpler one for yielding the energy, right? So whatever it is happening inside the cell, and the respiration is of cellular respiration. Okay. Now, one more important thing, if I will talk about the cellular respiration, what do you understand? It means that we are breaking down the bond. Okay, what we are doing, we are, the bond which is present in this carbon-carbon group, what we are doing, we are breaking down this macromolecule, and along with this breaking down of this macromolecule, we are yielding the energy in form of ATP. So ATP is what? ATP is the energy currency, or we can say that in which form they are resolving, in form of ATP, and ATP is what? Adenosine triphosphate. Okay, it is having 3 phosphate group, right? Now, when we are talking about the photosynthesis, it takes place where? In the mesophyll cells. When we are talking about this respiration, where it will take place? It will take place in the cytoplasm as well as the mitochondria. Okay, so if it is of any of the plant cell, okay, if it is any of the plant cell, we know that there is a particular cytoplasm. What it is there? Cytoplasm. It is also known as the cytosol, right? And we know that in each cell, what is there? There is a presence of what? There is a presence of different type of cell organelle, and one of the cell organelle, it is known as what? Mitochondria. What it is there? Mitochondria. Inside this, some other thing are also there, chloroplast is there, but on the green part. So here when I am talking about the cell or the plant cell, there is a cytoplasm as well as mitochondria. So in these two, there will be a complete oxidation of glucose molecule, right? So we can say that the cellular respiration takes place where? In the cytoplasm as well as the mitochondria. So here we will understand that how it takes place in the cytoplasm and how it will take place in the mitochondria, right? Now comes to the respiratory substrate. What do you understand by the respiratory substrate? It means that these are the compounds, okay, which we are going to break down. Okay, whatever we are going to break down for releasing the energy, it is known as a respiratory substrate. Why? Because on acting on this substrate, the plants are respiring. It means that they are taking the oxygen and breaking down their substrate into the proper form, as well as they are giving the CO2 also. Okay, so we can say these are the compounds that are oxidized during the respiration. Okay, and respiration is what? It is taking in what? Oxygen and releasing the CO2. Generally we know that in case of plants, the photosynthesis will be there, okay, during the daytime, because during the daytime sunlight is present, but at the night hours what happens? The plant gets respire. Okay, that's why it is said that we should not sleep under the tree. Why? During the night hours, because during the night hours they are releasing the CO2 and they are oxidizing their food, because whatever the food they have prepared during the daytime, they are utilizing that food during the night hours by oxidation of that food which has been stored in the plants, right here. So we will understand that how it will take place. Now comes to the ATP. ATP is, as I have said, it is the energy currency of any cell, because we are storing the energy in form of ATP. Okay. Now, during this respiration, what is happening? There will be exchange of gases. And what are the gases here? O2 as well as CO2. So what will happen in this gaseous exchange? There will be change of O2 as well as CO2, right? And in case of plants, we know that how the oxygen or the CO2 is getting inside this leaf with the help of stomata. Okay, if I will talk about the leaves, in leaves what are there? Stomata are present, and with the help of this stomata, what happens? The CO2 will enter inside the leaves, and due to this it will synthesize its food. So this stomata is responsible for the exchange of gases, right? For the exchange of O2 as well as CO2. So at the same time, we can say that these are responsible for entry of CO2 during the day hours, and during the night hours, these stomata are responsible for entry of the oxygen, right, and releasing the CO2. So what are the things or the components which are responsible for the exchange of gases? That is the stomata, and stomata, they are present on the green leaves, right? And what else other thing are there? We can say that exchange of gases takes place with the help of lenticels. Lenticels are what? They are the spaces or the pores which is present in the stems. Okay, you can see that a big tree, what happens, there are some spaces. Okay, these spaces are known as what? Lenticels. So with the help of these lenticels, again these stems, they are doing respiration. It means they are releasing the CO2, okay, and taking inside the oxygen. Okay, so here we have completed the basic thing about the respiration, how it will take place. So wholesome, we can say there is a oxidation of food, release of energy, and there must be a cytoplasm as well as mitochondria. So whole scenario will be in this thing, in this chapter. Okay, now comes to the respiration. Respiration can be of two types. Okay, generally we say that respiration is of two types. What are they? Aerobic respiration and anaerobic respiration. So aerobic respiration is what? The respiration which takes place in presence of oxygen. It means that oxygen must be present here. Okay, so oxygen will be there, and this type of respiration, it is known as the aerobic respiration. So the food which has been oxidized for this glucose, it will undergo in the aerobic respiration first, right? So whether there will be a absence or there will be presence. Okay, so there may be some slight changes. We will understand this, how it is happening. So aerobic is what? In presence of oxygen. In presence of oxygen, what is happening? There will be breakdown of this glucose, and during the breakdown of glucose, it will change into the pyruvic acid, and this pyruvic acid will again undergo into the Krebs cycle, right? So we can say the glucose, firstly glucose will break down, or there will be a partial breakdown. The complete oxidation will take place where? In the mitochondria, right? So in cytoplasm there will be a partial oxidation of this glucose, and in mitochondria there will be a fully oxidation of glucose, right? Now this aerobic respiration we have done. Then comes to the anaerobic respiration. See, if you will recall the primitive life on this earth, what happens during the time the oxygen content was not that much. So the organisms which are present here used to present on that time, what happens, there will be a less oxygen. So they have started living on that absence of oxygen or the lack of oxygen. Those are known as anaerobes. Anaerobes are what? Those organisms which can live in the absence of oxygen. They are known as anaerobes, right? So here we are having anaerobic respiration which takes place in absence of oxygen, right? So we have categorized this anaerobic respiration again into two types on the basis of their product. Okay, so the final product which we are getting here, on this basis we have divided this anaerobic respiration into two parts. What are they? Lactic acid fermentation as well as the alcoholic fermentation. And basically this anaerobic respiration is also known as the fermentation. Fermentation is what? When there will be some release of the we can say oxidation of food and release of CO2, right? So here what we are going to happen, if I will recall the whole process, we know that we have made this glucose during the photosynthesis. So now this plants will break down this glucose. So when glucose gets break down, it leads to several steps. Okay, these steps are known as what? Glycolysis. Glycolysis what? Glyco means the sugar and lysis means breakdown. So here the glucose which has been prepared by the photosynthesis, it gets breakdown here, and this glucose will change into the pyruvic acid. Okay, so this is the final product of the glycolysis. So if I will ask that what is the final product of the glycolysis, it will be the pyruvic acid. So whether it is a lactic acid fermentation or the alcoholic fermentation, okay, or it can be the aerobic respiration, the first thing will be common, that is the glycolysis. Okay, so the glycolysis step, we can say it is common for aerobic as well as the anaerobic respiration, and finally we are getting the pyruvic acid. Okay, so this pyruvic acid, what it will do? It will undergo it will undergo the reaction with the help of enzyme. Okay, so enzyme what we are using here, lactic acid dehydrogenase. Why? Because after this oxidation of this pyruvic acid, when it will undergo oxidation, it will form the lactic acid. Okay, what they are forming? Lactic acid. And for forming this lactic acid, which enzyme is required? Lactic acid dehydrogenase enzyme. Okay, and during this time there will be reduction and NADP and will change into the NADH2. Okay, so NADH is changing into the NADH2 here in case of lactic acid fermentation. If I will talk about the alcoholic fermentation, as I have said, glycolysis will be common for all respiration. So we are getting again pyruvic acid here also, and this pyruvic acid will undergo two steps. Okay, then we will get the ethanol, and ethanol is what? It is the alcohol. That's why we have said that is the alcoholic fermentation. So this type of fermentation you can see in examples like yeast. Okay, for the wine making, they are using? They are using the alcoholic fermentation, right? So during this alcoholic fermentation, the pyruvic acid, which is the 3 carbon compound, okay, 1, 2 and 3, it is a 3 carbon compound, and we know that glucose is a 6 carbon compound. So when this glucose will break down, it will break down into the 2 molecules of this 3 carbon compound, right here. So this pyruvic acid will undergo the decarboxylation process. And what is decarboxylation? It means that when there will be removal of CO2 from the reaction, it is known as decarboxylation, right? So here what is going to happen? This pyruvic acid will release its CO2 from here. Okay, when it will release the CO2, what it will form? It will form the acetaldehyde, right? And this acetaldehyde again undergo into the another step, and it will form the alcohol, and this alcohol is ethanol. And the enzyme which has been used here, that is pyruvic acid decarboxylase. Why? Because it is the pyruvic acid which is going to be decarboxylation, right? So for the decarboxylation, which enzyme is required? Decarboxylase enzyme. Okay, now comes to the another one, this ethanol. So for getting the ethanol, what is required? There must be a dehydrogenase enzyme which will change the aldehyde into acid. And generally we know that during the oxidation, alcohol changes into the aldehyde and aldehyde changes into the acid. Okay, so we have done this thing, alcohol to aldehyde, aldehyde to acid. So if we will go just reverse it, acid to aldehyde and aldehyde to alcohol. So this is about the fermentation, or we can say anaerobic respiration, right? In case of human beings, there is one more type of respiration which is known as lack of oxygen. Okay, so at that time or during the exercise, what happens? There will be a less amount of oxygen in our body. So that time there will be a formation of lactic acid, right? And that one we can say it is a lack of oxygen. So here majorly we have divided into two parts, that is the aerobic as well as the anaerobic respiration. Okay, now comes to the steps. What is respiration? If I will talk about the general definition of respiration, it means that breakdown of glucose. Okay, and during this breakdown there will be release of energy in form of ATP. Okay, so for happening this reaction, what is required? Two majorly steps are required, that is the glycolysis and Krebs cycle. Okay, glycolysis is what? Sugar will get breakdown, and there will be a 10-step process in the glycolysis, but still in this glycolysis there will be not completely oxidation of glucose. Okay, still in glycolysis, when it will get complete and it will form the pyruvic acid, it is a incomplete oxidation. So to complete this oxidation process, it will undergo the Krebs cycle. Okay, so in this Krebs cycle there will be a complete oxidation of glucose molecule. Okay, with the help of different enzymes. So in this chapter majorly we will discuss about the glycolysis and the Krebs cycle. Okay, and whatever the ATP we are getting, okay, whatever the ATP we can utilize directly, but during the Krebs cycle we will not get that much ATP. Okay, will not get ATP. So here what we will get here? NADH. Okay, so this NADH will give the energy when it will enter into the electron transport system. Right? So when we are getting the NADH, so we will send these NADH or FADH to where? Into the electron transportation system. From there what will happen? They will give the ATP, right? And from the glycolysis directly we are getting the ATP here. So now we will have to understand that what are the different steps which takes place in the glycolysis. Okay, so let's start with the another part. So now here we will start the first step, that is the glycolysis. As we have said, glycolysis is what? It is a Greek term. If you will split this term into two, that is a glycose. Glycose means sugar, and here we are talking about which sugar? The glucose, right? So here glucose as well as sugar, lysis means splitting or the breakdown, right? So what is going to happen in this glycolysis? There will be the oxidation or the breakdown of this sugar molecule, and due to this splitting, what will happen? It will form some different compounds. So we will study this glycolysis in 10 different steps. Okay, majorly here we are showing 9 steps. So what happens? Glycolysis takes place in a which type of fashion? Linear fashion, right? After the oxidation of this glucose, this is the first product, glucose, and the final product which we are getting after this glucose, it is a pyruvate. Okay, you can pronounce pyruvate, or you can also say the pyruvic acid. Both are the same thing, but still it is incomplete, and when it is incomplete, it will undergo another cycle, that will be the Krebs cycle. Okay, so in today's session we are not understanding the Krebs cycle, but here what will happen? When it will form the pyruvic acid, it will again enter into the another cycle, that is a Krebs cycle, and this one is a cyclic one, and here we can say it is a linear fashion, right? So now, as this glycolysis is given by 3 scientists, that is the Gustav Ammeden, Otto Meyerhof, and the J. Parnas, and that's why from these names we are getting EMP, and this glycolysis is also known as the EMP pathway. Why? Due to the contribution of these 3 different scientists here, right? These are some points which we have to memorize here. This is the EMP pathway, and this glycolysis, as I have discussed right now, that it is common both in aerobic as well as anaerobic. Okay, so starting from where? Glucose to pyruvic acid, and it depends that whether if it is a aerobic respiration, it will undergo into the Krebs cycle, but if it is an anaerobic respiration, it will not undergo this Krebs cycle. What will happen? There will be a breakdown of this pyruvate into the lactic acid as well as the alcohol. Okay, and for more important thing, the energy which we are getting from there, from the fermentation, it will be less energy. When either it is a lactic acid or the alcoholic, in both the cases we will not get that much energy. But when we will talk about the aerobic respiration, we will get more energy. Right here. Now comes to the another point, that where this glycolysis will take place. So glycolysis takes place where? In the cell. If it is a cell, any plant cell, so we can say that this is the cytoplasm. Okay, so in the cytoplasm, what will happen? The glucose which has been prepared after the process of photosynthesis, it will break down into this cytoplasm. Okay, so in this cytoplasm there will be the glycolysis. You have to memorize it, where it will take place? In the cytoplasm. And this Krebs cycle, which will takes place for the complete oxidation, it takes place where? In the mitochondria, right here. So we will understand first about the glycolysis. What are the different steps? We have started with the glucose, and in this glucose, what will happen? There will be a different step. So we have divided these steps here in very short. So we will understand these steps from here and correlate with this diagram, right? Now, the first thing which I have written here, the first step is phosphorylation. Okay, what do you understand by the phosphorylation? It means that addition of phosphate group. Okay, when we are adding phosphorus, we are adding with the help of which one? Phosphorylation. Okay, so if you can see here, this is glucose 6, glucose 1. Okay, one molecule of glucose is there, and we know that what is the formula? C6H12O6. So how many carbon compounds are there in the glucose? In one molecule of glucose, 6 carbon, right? So it is the 6 carbon compound. So we can represent it like this: 1, 2, 3, 4, 5, 6. Okay, this type of molecule it is there. So now in the first step, what we are doing? We are adding one phosphate group in this glucose. So when I will add this phosphate group in this one, what you will call it? Glucose 6-phosphate. Why? Because on the 6th position I have added this phosphorus group, and the process by which I will add this phosphorus group, it is known as the phosphorylation. And for this phosphorylation, what I am using here? I am using the enzyme hexokinase. So if I'm talking about that I have added, it means that this reaction has been done by the plant. So how the glucose will change into the glucose 6-phosphate? With the help of the phosphorus. So from where this phosphorus group will come? It will come from the ATP, because ATP is adenosine triphosphate. Okay, when it will get breakdown, it will give ADP as well as one inorganic phosphate. So this inorganic phosphate will add with this glucose and it will form the glucose 6-phosphate. Right here, and the enzymes which we have used here, hexokinase. Hexo relates to what? It means 6. It is a 6 carbon compound, and in 6 carbon compound we have formed this glucose 6-phosphate. So first step is phosphorylation, right? One more important thing which I have to discuss here, these are the different steps, right? If I will talk about this step up to here, okay, up to here, these steps are known as what? Preparatory phase. We have divided the glycolysis into two phases. What are they? Preparatory phase as well as the payoff phase. What does it mean? Preparatory means for the preparation what is required? We require the ATP. Okay, ATP is we are using from this step to this step, we are using the ATP, right, for addition of the phosphate group. But after this step, okay, after this one, what you will call it? Payoff phase. Why we are saying payoff phase? See here, in preparatory phase, they have done some job, and after doing some job, they are getting salary, right? So like the same way, in these steps, we are getting the salary, or we can say we are getting the energy, right? Up to here, we are using the energy, but here we are getting the energy, and this is the ultimate objective of the glycolysis or the respiration, that we will get the energy, right here. So what we can say, that in the first step we have used one ATP to form the glucose 6-phosphate, and it will change us into the ADP, right here. Now comes to the other step, glucose 6-phosphate as well as fructose 6-phosphate. If you will see the formula, it will be the same. Okay, that's why I have shown with the help of this reverse sign. Why? Because this glucose 6-phosphate can be changed into fructose 6-phosphate, and fructose can be changed into glucose 6-phosphate, right? So they are the isomers. What are they? They are the isomers of each other. So now what will happen in another step, this glucose 6-phosphate will change its we can say structure, but the formula will remain same. So glucose 6-phosphate will change into the fructose 6-phosphate, and here you can see that which step is their isomerization. It means that they will form its isomers. Okay, glucose 6-phosphate, it will form its isomer. So isomer is what? Fructose 6-phosphate, and for this isomerization there is no need of any energy. That's why we have not used any ATP here. Right here. Now, when we will get this fructose 6-phosphate with the help of which enzyme? Phosphohexose. Why? Because phospho is there, hexose means 6 carbon, isomer is why? Because we are doing the isomerization here. Right? Now, we will get this fructose 6-phosphate, which is also a 6 carbon. So, you can see that glucose 6-phosphate is also a 6 carbon. Okay, 1, 2, 3, 4, 5, 6. Right? And this one is also 1, 3, 3, 6. And 3, 6, right here. Now what you can see, that here up to here we said that only at the 6th position there is a phosphate group, whether it is a glucose 6-phosphate or the fructose 6-phosphate. Now in the another step again we will go for the phosphorylation. Okay, if you will see here, third step is again phosphorylation. Why? Again we are adding one phosphate group. We have added one phosphate group at the 6th position. Now in this step we are adding one more phosphate group at the 1st position. Okay, so the nomenclature will be like this. Fructose will remain same. Okay, and we have added one more phosphate group with the help of ATP here, and this will become the fructose 1,6-bisphosphate. It means that at 1st position as well as 6th position, it means at 2 position, okay, they are having 2 phosphate groups. So at 1st position and 6th position there is a phosphate group, and you will call it fructose 1,6-bisphosphate. Again it is a 6 carbon compound. Okay, so it is a 6 carbon compound right here. Now you can see that up to here we are having what? 3 carbon compound. Here phosphate group, here phosphate group. Right? Now in this next step what will happen? This will get breakdown. Okay? The carbon-carbon bond will get breakdown, and how it will break down? Into 2 molecules. Now Till here we are having one molecule of glucose, but from here we will get two molecules of 3 carbon compound. But here it was the 6 carbon compound. When we will break down this bond, what we will get it here? We will get two molecules of triose phosphate. It means that phosphate is also present with this trio, and phosphate is also present in this trio. So this trio is what? 3 carbon compound, and this is also a 3 carbon compound, and both are what? They are isomers of each other, right? So here what we can say, that we are getting triose phosphate. Triose phosphate is nothing, it is a 3 carbon compound which is having the phosphate group. Okay, so I have written here 3 carbon compound. Okay, then it can form the glyceraldehyde 3 phosphate, or the, we can say, dihydroxyacetone phosphate. Both are having the same type of formula. So what we will get here? We will get the two molecules. Now whatever the things we will follow here, we will follow for the two molecules. Up to here we are having only one glucose molecules, right here, but we will study according to the one molecule. Later on we will convert into the two molecules of this triose compound, right? So here you can see that from here where it will get split down, so it will form 3 carbon compound, and this 3 carbon compound is what? Glyceraldehyde 3 phosphate. In short we are saying PGAL, and PGAL is what we can say, phosphoglyceraldehyde. Okay, so what it is? We can say phosphoglyceraldehyde also, we can say glyceraldehyde 3 phosphate also, because at the third position there is a what? Phosphate group. Now when we are having this 3 carbon compound, okay, again it will divide, again it will form what? As I have said, alcohol changes into aldehyde, aldehyde changes into acid. So what you can see is, this is the aldehyde, this aldehyde will change into what? It will change into the acid. So what it will form? Here it is a glyceraldehyde 3 phosphate. Now you will get what? You will get the acid. Here two steps may be there. Okay, directly it will form the acid, then it will attach the phosphorus group. But here we have done both the same thing in the same step. That's why it is a 9th step. Right? So what you have to understand, that this glyceraldehyde 3 phosphate, what it is doing? First thing, it is forming its acid. Second thing, it is adding one more phosphorus group. Okay, so if it is a 3 carbon compound, if it is a 3 carbon, already it is having one phosphate group. Okay, now what we want? We want that there must be a one more phosphate group. Okay, there must be a one more phosphate group. So what we have done? We have added this phosphorus group. Okay, as well as we have converted into the acid. So what it will become? It will become the 1,3-bis phospho, and we have converted in this aldehyde into acid. So it will form the glyceric acid. So what it is there? 1,3-bis phosphoglyceric acid, right? And here it is what? It is the again 3 carbon compound. Okay, you can see that again it is a 3 carbon compound. Now in the another step what we are doing? We are removing this phosphorus. Firstly we have added the phosphorus group for what? For forming this acid. But in the next step we are removing this phosphate group from the first position. And when we will do the same thing, what it will form? It will form the 3-phosphoglyceric acid. Okay, already it has become the glyceric acid. So we have removed one phosphate group, and it will form the 3-phosphoglyceric acid. And the enzyme which has been used here, it is the phosphoglycerate kinase. Phosphoglycerate kinase is what? When we are removing the phosphate group. Okay, and listen here, up to here we are adding the phosphate group. That's why we have used the ATP. But from here what you can observe? You can observe that we have not used the phosphor. Okay, we have not added the phosphate group. So what we have done? We have used the ADP, and this phosphate group from here, this phosphate group which has been placed here, it will break down and it will attach with this ADP. Okay, when it will attach with this ADP, what it will form? It will form the ATP, right? So what we are getting here? We are getting the energy. That's why we said it is a payoff phase, and before that it is a preparatory phase where we have used the ATP, and here we are forming the ATP, and this is the ultimate reason for the respiration, that we should get the ATP from the oxidation of food, right here. As far as you can see, this NAD will also get converted into the NADH2, right? So these are also the energy sources, but what happens in the NAD to NADH2, here it has to go to the electron transport system, then only it will give the ATP. But the ATP which we are getting from here, we can utilize this ATP directly. Okay, so that's why this process in which we are getting directly ATP, it is known as the substrate level phosphorylation. What does it mean? That whatever the phosphorus group is present here, we have oxidized directly and it will not undergo any of the electron transport system. Okay, so how much step we have completed here? We have done the phosphorylation. Okay, then we have done the splitting also. Then we have done this conversion of phosphoglyceraldehyde to the bis-phosphoglyceraldehyde, from where you can see here this phosphoglyceraldehyde is present here and it will change into the BPGA. BPGA is what? Bis-phosphoglyceric acid. Then this BPGA will again convert into the 3-phosphoglyceric acid. Okay, and the enzyme is what? Phosphoglycerate kinase. Then up to here it is a 3-carbon compound. What it is? 3-carbon compound. Now what we are doing? We are removing one more carbon group. Okay, and with the help of which enzyme? Phosphoglyceromutase. With the help of this enzyme, what we are doing? We are removing one carbon molecule, and due to this, what it will form? 2-phosphoglycerate. It means that now it is only the 2-phosphoglycerate, right? Now this 2-phosphoglycerate again changes into the phosphoenol pyruvate. Okay, or we can say phosphoenol pyruvic acid. So what it is? It is phosphoenol pyruvate acid, or we can say pyruvate. So this phosphoglycerate will change into the phosphoenol pyruvate with the help of enolase enzyme, right? And again it is a interconvertible. This is the reversible process, and with the help of these cofactor, that is a magnesium ion. Now again when this 2-phosphoenol pyruvic acid, which is again we can say a 3-carbon compound, because up to here what we are initially it was a 6-carbon compound, then we have broken down into the 2 molecules of the 3-carbon compound. So up to here what we are getting? We are getting the pyruvate. So pyruvate is what? It is a 3-carbon compound which we are getting from this phosphoenol pyruvate acid, and this phosphoenol pyruvic acid will form the pyruvic acid, and this is the final product of the glycolysis. So what we can say here, or we can sum up from here, that we have used ATP here. So how many ATP you have used here? 1, 2. Okay, but how many ATP you are getting from this glycolysis? You are getting 1 ATP from here and 1 ATP from here. Okay, we are getting 2 ATP, but it is what? It is a 2 molecules, so we can just multiply it 4 ATP right here. But again this is a direct ATP which we are getting from this one. This NAD, always NADH2 as well as FADH2. NADH2, FADH2, they are also giving the ATP, but NADH2 gives 3 ATP molecule from each NADH2, and from one FADH2 we are getting 2 ATP after the entry of the electron transportation system, right? So we can say that directly we are getting how many ATP here? We are getting the 4 ATP, right? And out of this 4 ATP, already we have utilized these 2 ATP in this glycolysis, right? So this is all about the glycolysis which takes place in the cytoplasm of the cell. Now in the another class we will study that this pyruvic acid again enter into the Krebs cycle and how it will complete its oxidation. Okay, so that's all for today. Thank you.